Well, first of all, I would like you to not get me wrong. I mean, it's not that I'm questioning the value of all the tremendous progress that are made in the field of phenotyping, but I think it's as technology develops, we may sort of slip in the habit of measuring everything because the technology allows it, and then run into problems of what do we do with the data. And so this morning, what I want to do is to take you a bit through the work that we do on drought, because I mean, you can measure a lot of things on drought, but it's really what matters for drought adaptations. And so I want to take you through a bit of the work that we do on drought in my group, and what sort of questions we came up with, and what sort of question, how to design platforms to answer those questions. So there's 5 sections in my presentation. The first one is basic consideration around trait phenotyping, and then the 4 following sections are different sort of domains of phenotyping that we do for drought adaptations. And in each case, what I'll try to do is what we do and where we arrived, and what sort of challenge we've got on the phenotyping side, and what sort of platform we need to answer those questions. So that slide shows the AppSim generic crop template. AppSim is a crop simulation model. It predicts yields based on the weather data. And so what you've got here is basically the grain yield. And what you've got to understand is grain yield is really sort of a lot of building blocks coming up to yield. And we always need to keep this in mind, that you've got yield on one side and you've got the mechanisms that take you to yield. So if you work on any of those building blocks, you need not forget yield. If you work on yield, if you're a breeder, for example, you need to understand really clearly that there's building blocks underneath. And of course, with new technology for phenotyping, where we probably should focus on those building blocks. When we do phenotyping, there's 2 issues I think we need to sort of also be very clear with. The first one is, is my phenotype right? So what I'm measuring, is it really what I need to measure the level of adaptation of plant to drought? And so there's 2 cases. You've got an accurate phenotype, so you're right on target, or you are not accurate. That means that you measure some things which is probably not the right phenotype. And then the second question, once you have the right phenotype, is the measurement of that phenotype good enough? And so you've got 2 cases. You are very precise in your measurements, so it's quite important for breeding, because for breeders it's a matter of heritability. So if you are very precise, you increase your heritability. If you are not very precise, then you've got less heritability, and then you've got less chance to breed for a particular trait. And then if you are not precise, which means that you need to develop methods to increase the level of precision. And so as long as you are clear on those ideas, then you can get going. Then what is a drought-tolerant plant? I mean, we've talked already quite a bit in that conference on what is drought tolerance. Yesterday, I queried whether we talk about drought tolerance or survival. On a crop production perspective, I think we should forget about survival, but really keep in mind that we need to have a yield eventually. So a couple of hypotheses around which we have framed the work in the lab is a drought-adapted plant, a drought-tolerant plant, inverted comma, is a plant that essentially has enough water to fill up the grain, but then has no longer any drop of water in the soil profile at the end of the crop cycle. So in other words, it is the idea that a successful crop under water-limited conditions maximize water extraction from the soil, but maximize it and at the same time manage a limited amount of water in a way that it makes it to the finish line. So there's really 2 domains of work where we work. The first one is really tapping water and maximizing the water capture. And the second one is how the plant, through different traits, manage that water budget. And so the focus on our traits is really traits that touch on the water budget of the plant. So the first part of the talk is what we do on the root side. Well, I've presented that last year at Phenodays 2012. That's the lithometric facilities that I've developed at ICRISAT, which is a set of long and large tubes. Well, in that one here, those one are 20 centimeters in diameter and 1 meter 20. We operate at a planting density of about 20 plants per square meter. So we want to really reproduce the planting densities that we've got in the field environment. And those one here are 25 centimeters, 2 meter long, and we operate at a planting density of about 10 plants per square meter. The whole idea with this is really to reproduce as close as we can the field conditions. We are outside, we measure yield, very relevant yield data, but at the same time we weigh those tubes, and the differences in weighing, consecutive weighings, gives us plant transpiration. So we are capable of measuring all the water budget of the plant throughout the entire cropping cycle. And well, we've got fairly high throughput and a fairly large capacity with those cylinders. So with those cylinders, we could also extract the roots and measure the root length density. And there's a lot of work on the roots that look at really the morphological parameters of roots. Well, in that slide, you've got root length density here in the x-axis. I give the example of two crops, beans and chickpeas. And in the y-axis, we've got water use. And you see that there's basically no relationship between both. And I've got data on other crops. And so, in other words, the take-home message from that slide is measuring root length density is really not the right phenotype. And on top of it, it's not very precise. So we are really off range and with a non-precise phenotype. The second thing we did with those tubes was, well, if we have crops that ask to maximize water use, well, we are going to measure the total water use and the drought stress conditions of those crops in those cylinders. And so if you've got the pod yield in the y-axis as a function of the total water extracted, we did that measurement across different crops. It gives an example here of 2 seasons of peanut data, beans, cowpea. As you can see, same thing. There's no relationship between the total water extracted from the soil profile and the pod yield that we get under terminal stress conditions. And we got very similar data also in cereals. So here we have a phenotype which is very precise, but again, it's off target. It's not the phenotype that we need to look at. So it's really not about the total water that's extracted from the soil profile. So what we did was to measure the kinetics of water extraction. So what you've got here is the water extracted as a function of time after sowing in a set of 12 tolerant and 8 sensitive chickpea genotypes. They were selected based on field assessment and the terminal stress. What you can see, the green dashed here are the mean of the tolerant materials. They extract less water at a vegetative stage, and then they sort of catch up on the sensitive one during and after flowering time. And if you do a bit of regression analysis between the water extracted here at the vegetative stage, here in the x-axis, and grain yield, you've got a negative relationship. So the more water used before flowering, the less yield. And then if you correlate the water extracted during and after flowering and grain yield, you've got significant positive relationships. And in fact, in the end, you don't have major differences in the total water extracted between those tolerant and sensitive lines. It's really how they have used a limited amount of water and how they have managed that water. So what are the hypotheses? Well, if we have a lower water use at vegetative stage, it could be because of smaller canopy development or lower leaf conductance. Here I want to show you similar sort of data in pearl millet. Again, that's the water extraction as a function of time after panicle emergence. The last irrigation is given here at about booting, and two sensitive material extract more water around and before flowering at a time when there's still no stress, but then they sort of run short of water later on. The tolerant material tend to extract less, and then they are left with a bit more water later on. If you sum up all those data points, there's no differences in the total water extracted from the soil profile. It's again, like in the case of chickpeas, it's again a difference in how the plant has managed a limited amount of water. And if you look at the water extracted in probably the critical weeks, 3 weeks after panicle emergence, so it's about maybe a liter and a half of difference, and maybe another half liter here. If you've got 10 plants per square meter, here we talk about no more than 15 or 20 millimeter of water, which is really not much, but it's very important. And thinking of yield, well, if I correlate that water extraction, water use in week 3 after panicle emergence and grain yield, we've got a very significant positive relationship. So we are we have a very right phenotype, right on target, on which we could sort of improve the precision. So what platform do we need for phenotyping if you want to look at root traits? Well, that platform is already developed. It's outside. It's yield-based. And what we need really to focus on in water extraction at critical time. What we are working on at the moment is really finding ways to automate the weighing, for example, by having cylinders sitting on top of load cells. Of course, if you've got more water extracted during reproduction, well, you could infer a different phenotype. You could measure it from, for example, canopy temperature, because if you've got temperature, you should have a cooler canopy. But as you can see in that slide here, when you've got more water extracted in the grain filling period, then you end up with a stay green phenotype. So that's also the sort of thing we could phenotype. What is important to note here is either a cooler canopy or a stay green phenotype. Well, it's nice to phenotype. You can do that maybe in the field, but it's only going to be consequences of other mechanisms and traits that have operated before. So it's important to, and yesterday I think someone was, or this morning in fact, someone was talking about the early phenotype and the late phenotype. Well, so the stay green and the canopy would be like what was called late phenotype. And then we are going to talk now about what are really the founding factors, the early phenotype. So let's talk now about leaf area development. Here is a typical curve showing the leaf area development as a function of thermal time in sort of hypothetical genotypes. In the dashed line, you've got those that have early vigor, and the solid lines are those that have sort of slow early vigor. But then you've got some that end up with a low leaf area or a high leaf area. The differences in the surface area between those curves, for me, physiologists working on drought means water. So in other words, a line like this one would use less water, and a line like this one would use more water. This one would use more water here and less water here. So we have looked at that and the dynamic of development, because it's, of course, going to be affecting a lot. How much water is needed to reach flowering. So here is reflection of that in, for example, sensitive peanut, tolerant peanuts. That's water extraction as a function of time. That's measured under fully irrigated conditions. The tolerant line, use less water to develop the canopy, and it gets reflected in how much water it consumes. Sensitive material use more water, or more bushes, or they are more leafy. And so what we do now in the field is really to sort of measure those development coefficients, or those exponential coefficients linking leaf area and known number on the main stem. And what we measure are those exponential coefficients and the range of genetic variation that we've got. What sort of platform do we need for that is, of course, something that can dynamically measure the leaf area over time. If I get back to that, the example of chickpea that you've seen before, so if we want to look at genetic variation for low early vigor, so we do that in the field. Again, that's leaf area as a function of known number in the main stem, and we are basically sort of exploring the range of genetic variation for those traits. A bit of work in sorghum now. That's again the leaf area at anthesis. We've seen that we are working on stay green. We have material introgress with stay green QTL. We have shown that we've got a major effect of the stay green QTL on the reduction or the alteration, let's say, of the leaf area. And here is a bit of regression between the grain yield and the terminal stress as a function of the leaf area that was measured at anthesis. We did that in those tubes. At that stage, we did that manually. You see that's a clear negative relationship. So those that are growing too larger canopy basically run short of water during the grain filling. And so what, again, in the case of sorghum, we look at basically the dynamic of leaf area development. Here in the y-axis as a function of thermal time. And the well-watered condition, you see all the range of variation. And then there's one more factor we need to sort of take into account, is the effect of the vapor pressure deficit on the leaf area development. When it comes under drought stress, we've got, of course, a reduction in the leaf area that gets developed, but it doesn't get reduced proportionally the same in all genotypes. So there's really two factors that we need to sort of look at if we really want to capture the genetics of leaf area development. What is the inherent sort of properties, and how it responds to VPD, how it responds to soil moisture. So there are two coefficients we measure here. Is really what we call the total plant production coefficient, and here an inflection ratio that would reflect basically the speed of early development of the canopy. With those coefficients, after that we can assess, what is the effect they would have using the crop simulation model, APSIM. Forget about the sort of complexity of that slide. Just follow LAI, the leaf area index. The red one is a material that has developed a large leaf area. The blue one is a material that has developed a slightly smaller leaf area at anthesis. And that's basically senescing occurring a lot quicker in the plant that developed a larger canopy. And there's a delayed senescence, which end up, in fact, with a stay green phenotype. A bit like what we've seen before in pearl millet. So again, stay green as a sort of late trait, explained by earlier trait dealing with leaf area development. So what sort of platform we need for that? Well, again, it's a matter of measuring quicker in vivo leaf area and the dynamic of leaf area development so that we can infer those development coefficients. But of course, the platform needs to be designed in a way that we can also test the effect of water stress. So we need to be able to measure transpiration, measure the level of stress, measure the soil moisture in our growing conditions. But we also need to be able of measuring the effect of VPD. So it means that, well, if we work on closed chamber, or if we work well, not in closed chamber, if we work in glass house, where we're going to have problem to test the effect of high VPD, the condition that we face in our semi-arid conditions. And to measure the VPD effect, the way we are going to be doing is to work outside during time of the year when the plant are challenged by those VPD conditions, but then we need to have a very, very high throughput so that we can measure leaf area development under low VPD conditions and under high VPD conditions. So it's all sort of thought that we have to put to develop the right platform to phenotype for those critical traits. Next section, leaf conductance. Well, here is again, a sort of schematical representation of transpiration as a function of vapor pressure deficit. Typical response, you increase the evaporative demand or the vapor pressure deficit if you prefer. You've got a linear response. But then across different genotype and different species, well, you've got all sorts of response. For example, you've got this one, which has the same response as the solid line, but gets sort of a breakpoint when it means a high VPD. So it means that it restricts the transpiration of the high VPD. That one here saves water here by having a lower leaf conductance under low VPD conditions, but further restricts transpiration in the high VPD. And then you've got those cases where, the lines are not sensitive to VPD, but they've got a very, very small slope of response to VPD. Again, it's like in the leaf area development case, the differences between those curves for us means water saving. Okay? And so what we are phenotyping for is for that trait, because we've seen repeatedly across different crops that it has, a strong bearing on drought adaptation. So here is an example of that transpiration response to VPD in the case of pearl millet, terminal drought sensitive material, unrestricted at all, terminal drought tolerant, differences in conductance under low VPD, first mechanisms of water saving, second mechanisms of water saving, a further restriction of the transpiration under high VPD conditions. So we want to phenotype for those two traits because they are, well, first of all, it's a very convenient, very precise phenotype, and it's of course right on target. In fact, it's probably the main trait explaining those differences in the kinetics of water use from the soil profile that I've presented in the second part of my talk. So we want to phenotype for those traits. Well, so far, what have we done? Well, we have simply done a good old way by measuring leaf transpiration in plants that are grown in pots. All that is measured under fully irrigated conditions, by the way. So it's done destructively. And of course, for us, what we need now is a quicker way to assess leaf area, is a quicker way to measure plant transpiration, and we are developing a platform that would allow us to do that quicker. But if you don't have the platform, here it's really not about the platform. It's really about what we want to measure. And here we designed the experiment in a way. We wanted to measure transpiration rate and the low VPD by weighing at the time of the year, at the time of the day when the VPD is low, and we wanted to measure the transpiration rate and the high VPD, and we did that at the right time of the day when the VPD is what we need to be. So it's really not about the fanciness of the platform. It's really how you would use a platform to measure exactly the same thing. And last year, I think I've presented the mapping of that work. Here now, a bit of data on that VPD response in different crops. So here is a case of peanut. We've got some variation in the response. Here what I want to show you is, so we have genetic variation for the transpiration restriction and the high VPD in the case of peanuts. And what we are doing also is a bit of simulation modeling to evaluate the effect that trait could have on yield across target regions. So here is a block of West African countries, and the color coding is basically denoting the yield benefit in percentage if you have that trait bred into your material. And if it's very blue, it means that you've got a very, very strong benefit, and that would be the case, especially in those locations. So it's not only about looking at what is your accurate trait, but it's also then after about testing the effect of that trait and the validity of that trait across different regions. A case of cowpea here, where we have also differences in that response to VPD in tolerant material, again tested under field conditions. So a breakpoint and water saving in the tolerant, no breakpoint in the sensitive. So there also we phenotype, the good old way, by measuring leaf area and the transpiration response. Well, since we are interested in the water budget, we wanted to know in the recombinant inbred line population of cowpeas what drives transpiration. And so as you can see, transpiration here in the y-axis is explained about 70% by the differences in leaf area. Okay, so no surprise. But at the same level of leaf area, you see that you still have quite a bit of variation in the transpiration. And so that residual variation is explained by the conductance and the high VPD. So these are the two phenotypes, the right one, that we phenotype for and then mapped QTL. So we've got QTL here for the transpiration rate, for the leaf area, given by one of the two parents, and then here QTL for the response to the VPD given by the other parent. So there, we've got a clear understanding of what of the two parents, what alleles each of those two parents is bringing in the water budget. And so it's quite a sort of powerful tool. So what platform do we need for that is, again, the platform that measures leaf area very quick, but leaf area plus the transpiration measurement and the low and high VPD measurements. And then we need to test, of course, the VPD effects. The last section of my talk is going to be very short. It's growth response to soil drying. Well, here is, again, a typical response of transpiration, which we take as a simple proxy for growth, or for photosynthesis, if you want, as a function of soil moisture. Typically, plant sort of start restricting growth when about 60% of the soil moisture has disappeared. So which means that under drought stress conditions, it's probably at least equally important to look at what happens before there's any stress than focusing only at what happens when the plant is under stress indeed. What is quite important with this is we've got genetic variation for the moisture threshold where transpiration drops. So you've got those, sort of the careful guys, and that start declining transpiration at very high moisture threshold, and those that are like opportunistic and then keep on transpiring at the same rate as a fully irrigated plant until there's almost no more water in the soil profile. So basically, on our standpoint, early growth decline means water saving. But of course, drought varies. If you've got a rain or an irrigation here, so this genotype that transpires full blast until here would really sort of lose nothing by keeping on transpiring late, while this one would probably sort of be more, would have a penalty in the yield by having sort of being too conservative in its water use. So it's always those things that we need to be very clear. I mean, there's no drought tolerance. There's only trade that touch on the water budget one way or another, and then it's up to us to sort of fit them back in a context, a drought context, and to fit them to the best possible way. Bit of a genetic variation we've got for that trait. Here's a case of cowpeas. Here is a case of chickpeas. So it's again a phenotype which is really right on target. What sort of platform we need for that? I mean, those platforms exist already. I mean, the Lambda facility, the Plants Accelerator is a fantastic platform for that. Nobody uses it for that purpose. Too bad. Because we could have a really sort of high throughput measurement of those traits if the protocol were designed properly in the use. So in summary, yeah, no doubt. I mean, there's great opportunities with the new phenotyping platform. The technology has advanced. I mean, in a way that's, really fabulous. But I think we are at a stage where we need to sort of, sit back and sort of ask questions. And it's especially true for the case of drought. It's extremely complex. It's nice to have 8 features sort of correlating with a simple trait. It's going to be a bit more complicated with, a constraint like drought, which is so complex. I think we need really to focus on the building blocks, and those building blocks, I think, revolve around, simply the water budget. So, building blocks, we need to sort of split them down in, the late trait and the early trait, and the more we go towards the founding factors, the early traits, I mean, the better, as long as we know that they are right on target. Important to always keep in mind the relation to yield, so that's why my orientation is really to deal with the environmental variations, the best we can, and be outside the best, the more we can. And of course, in those assessment of traits, those building blocks, one of the big things that we'll have to deal with is really the response of traits to environmental cues. Because we measure traits, we think they are like fixed in stone. And actually, in a lot of cases, what we measure is really responses to environmental conditions that vary. And so it's really not until we have a better understanding, a better ecophysiological understanding of those building blocks that we can really make a dent. And those 8 features are nice, but there's a layer of understanding we need to add to that sort of approach. I'll leave it for now.